Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hello, everyone, and welcome back to the Genetics Podcast. I'm excited to be here with one of my good friends and definitely the most featured guest on the podcast so far, Dr. Veera Rajagopal. We met years ago and did initially a yearly roundup of some of the most exciting stories, but we decided to try doing it every 3 months this year because there's just too much good stuff to talk about at the end of the year. So, Veera, welcome back, and thanks for taking the time.
Thanks, Patrick, for the invitation. Happy to be back again.
We've got 4 themes to talk about today, 4 papers that are part of some new themes, but also some long-standing themes. I wonder if you could kick off the first one, which is my favorite, just because I spent so much of my PhD work looking at noncoding regions and their role in developmental disorders. And this was just an amazing story, an amazing piece of work that has directly resulted in diagnoses for now hundreds and probably soon thousands of families. So I wonder if you could talk about this one, because it's amazing stuff.
Yeah, so before I start, I should tell there's so many papers got published in the time before, since we did our last recording. So it's just like I'm choosing, with no any particular criteria, just probably if I just found this most interesting, and there are really interesting biology and how the genetic discoveries evolve over time. So, but yeah, I just want to emphasize that there's tons of paper out there and we can just sit and talk for the whole day.
Yes.
And yeah, the first paper, I think this definitely comes as the top one of all the papers and probably is one of the most important discovery in this year, I would say. So the discovery here is that there are researchers from University of Oxford and also from the Broad Institute and in collaboration with Genomics England. So they found one recurrent de novo mutation that is as one of the most common cause of neurodevelopmental disorder. We've been covering on the topic of whole genome sequencing in our past recordings, right? So what new things we will discover as the whole genome sequencing picks up, right? So we are doing it in the population scale, focusing on healthy population, but also there is on the other side, whole genome sequencing is we are realizing the importance in genetic diagnosis for rare diseases, and this incredible resource of the 100,000 genome project from the Genomics England has sequenced these many patients, rare disease patients, right? So I always mentioned, the non-coding genome is really a big space, and the probably the initial major insights come from very highly penetrant Mendelian conditions, and we have seen it again and again. And this is another interesting discovery, but probably very important discovery. So they identified one de novo mutation. This mutation is located within a small nuclear RNA gene. And so the incredible aspect of this is it turned out to be extraordinarily common. They analyzed the a set of NDD patients among the Genomics England. I think around 3,800 or so patients who don't have a genetic diagnosis yet. So a lot of researchers are working on this data set to identify the genetic diagnosis, right? And so in this analysis, they identified this one particular de novo mutation within the small nuclear RNA gene, and it turned out to be very common. And it's a de novo mutation, so it's kind of recurring again and again. That's one of the most fascinating aspect. But but also need to understand the impact of this, because de novo mutations are very, very rare, right? So they are like, just de novo. So the chances for occurring is like extraordinarily rare. So not often we see that the same mutation causing, like the same disease again and again. So this is one particular example. And it turned out, this is even more common than the most common cause we would have ever seen before. And so we always talk about focus on coding regions, right? So all the past decades of genetic diagnosis all has been focused on coding regions, because we thought that, like we always assume that most of the causes reside over there.
Yeah.
And this is kind of a finding. This is like an incredible finding that comes and, pats us back on the back of our head saying like, see, these are the examples. So we missed it. It's a pity, right? I mean, so the prevalence of this mutation estimate is like 0.5 percentage of all NDD cases. So imagine how much this would scale across the world. So probably thousands, or even, like many tens of thousands of patients across the world can be explained in NDD patients can be explained by this particular mutation. So that's the, like really very fantastic discovery. So what exactly is this variant? It's an insertion variant. It's located within a small nuclear RNA gene. And so they found this region around this mutation in this snRNA gene is like very constrained. So I think we discussed about the whole genome sequencing data from the UK Biobank. One of the important use of this having a whole genome sequencing data, the population scale, is like we have mutation constraint estimates for genes. Now we can have mutation constraint estimates for the non-coding regions. And that is one of the, one of the important findings from the whole genome paper from the Decode initial flagship paper came is like there are regions non-coding genome that are even more constrained than actually the coding regions, and probably there are mutations sitting over there, that are causing neurodevelopment disorder. And this is one example. So there is an 18 base pair region around this mutation in this snRNA. It's very constrained, like in the UK Biobank whole genome sequencing data. So basically, first they identified this one mutation. It is a 6465, an insertion, T insertion between these two nucleotides. And that is the most recurrent one. But then they studied, okay, this region, and they found this region is highly, constrained. Like there are very few mutations than what you would expect, based on the different metrics. And then they looked for other variants in this gene, and they further found another secondary insertion variant that is also seen in, like a handful of patients. And there's also other SNVs. All of these patients have remarkably very similar neurodevelopmental phenotypes. They have like developmental delay, motor and fine, like cognition skills affected, and also speech, and also they have epilepsy, short stature, microcephaly. So a lot of these features are incredibly overlapping across all these patients carrying this mutation. So it seems like one of the important neurodevelopmental syndrome that we've been not aware of all these years, And now already I think like since this paper was published, like I think like a lot of people already reached out to Nicola Wiffen, who is the senior author in this paper from University of Oxford, that's saying, hey, we already, we find this mutation in our samples. So people are already reaching out. So many people are already finding it, and it's going to be, still we'll be hearing a lot of these stories. And like the bottom line is like there are so many parents are going to find the reason for their neurodevelopmental condition in their children. And so there is like two aspects of it that is like really interesting to discuss. One is the biology. I mean, like what exactly is happening, why this mutation is causing a neurodevelopmental disorder. So this is present in a small nuclear RNA gene. And small nuclear RNA is an U4 snRNA. So these small nuclear RNAs are a component of a spliceosome complex. So this spliceosome complex is important for all the splicing, right? So the removing the introns, stitching the exons together. So it's a fundamental, biology of in biology, right? So that we already know that like some of the mutations in this proteins involved in this spliceosome complex causes neurodevelopmental disorder. And so exactly how this is causing the like the affecting the neurodevelopment is still not clear. I think like they even looked at blood sample of some of these patients, looking at, if it is affecting the spliceosome complex, then you would see this aberrant splicing defects in the, like in the blood. If you look at the RNA expression, but they couldn't find anything. It's clear that, it's having like a dramatic impact on the brain development, and it is highly expressed in the brain. But it'll be really, interesting to see how this turns out. What is the mechanism here? But the other interesting aspect of this is like the extraordinary recurrence, right? So unbelievably common,
Yeah.
this mutation. So there is a strong purifying selection. So this 18 base pair region is devoid of mutations. Whatever new mutations occurring in this region is being removed again and again by purifying selection, natural selection. Then there's a new mutation appearing here. So there is one of the interesting aspect is like there is recurrence of the mutation. And the second interesting aspect is like specifically the recurrence of an insertion mutation. So it's extremely, like impressive, like how insertion mutation is becoming is observed very commonly. Now we have like 2 recurrent insertion mutations. So even though we don't know like what is the exact mechanism, I think the authors gives us like 3 theories. Like one is that this is an open chromatin region. So the open chromatin region are accessible to all mutagen proteins. So there is a very high chance for, undergoing, like mutagenesis. But even that cannot explain the insertion mutation. So probably, like the idea is that, so this is an RNA gene, and the sequences are often, prone to form secondary structures. So this secondary structure formation can actually leads to, like the mutation mechanism where you end up inserting a nucleotide in the, in the DNA. So the third explanation is this: probably there is some kind of a germline selection, particularly in the, like in the spermatozoas. And so that it could give some kind of an advantage, so that results in the sperms carrying this mutation having a selective advantage to, fertilize the ovum. So we know those kind of examples, but it's not clear if this also gives us kind of an advantage. Probably someone will be doing sperm sequencing, germ cell sequencing, to see if it is the case. But it's really, interesting.
I don't know if this is overlapping with one of yours, but we can check with Nikki if we can include this. But I've heard that someone reached out to her on Twitter and pointed her to Nikki is the senior author on the paper, pointed her to a paper from Shamil Sunyev's group that looked at RNA polymerase 3, and it has a mutagenicity discovery that's been made recently. And so there may be a link there between RNA polymerase 3 and a specific mutation signature that it has that that matches what they're seeing in these regions. So it could be kind of a very niche kind of hypermutator phenotype that's happening here that happens to kind of break the small non-coding RNA in a specific way that causes this pathogenic phenotype.
That's very interesting. Yeah. So, I mean, finally, the therapeutic implications, right? So obviously, this results in a, diagnosis for thousands of patients. Already it's happening. It's a big impact, translational impact, the kind of impact we really, want to see in kind of these genetic discoveries. And the other thing is going to be the companies, or, like many people are now going to want to study this mutation because such a big target population, thousands of patients. I don't think we have any kind of such, very few like MECP2, Rett syndrome. So there's like very few diseases where you have like very commonly occurring mutations. So probably there's a big interest in, finding out way to treat this condition because of a big target population. So this is like a beginning of a, long evolving story. We will hear more and more about in the upcoming years, like more interesting aspects of this, this neurodevelopmental disease. So I think this is something like a disease that everyone should be aware of, like is going to be very commonly known across the world, at least across the clinical genomic community.
Yeah, and I wonder, this is a total prediction that I may regret making, but this seems like it could be a candidate for fastest time from discovery of the disease to treatment of the disease, because it's discovered, very recently, but it feels like it has a lot of attractive properties. I mean, it has the unattractive and challenging property of being a severe developmental disorder in children, but obviously, for good reason that makes it a super important area to go after. But The fact that it's the same change in all of these families means that there's a lot of things going well for this population, and that there may be a very precise targeted treatment that is applicable to a very large population. So I'm interested to see how the drug development, treatment development side of this progresses, because like you said, it's a very attractive target for a lot of reasons.
I mean, like, it's interesting you mentioned that, because one of the common discussions we have is like how much time it takes from genetic
Yeah.
discovery to, like clinical developing a drug, right? And it's on average is 25 years. It can be, there are some for which you have even like 50 years. But this is like the timing of the discovery is also important, right? So right now, technology is advancing in a very rapid scale. So even, if something is newly discovered, we already have all the technologies and modalities in place to, get into action and, like try the drug, develop the drug, and, like run the clinical trials. So your prediction might be good, correct, because of the current phase, probably we will soon see like drug candidates and clinical trials. But this is not the case for, like monogenic conditions or conditions that's been discovered 10 years ago or 20 years ago. And so we should also think about that when we actually, people blindly compare, like take an average of how much time is taken from genetic discovery to drug development. But the timing is really, really important, because when the discovery happened, what kind of technologies that we had at our disposal to treat the conditions. So now it's going to be fast, because so many things are happening, CRISPR, gene therapy. Yeah.
Yeah, and I think it'll be one of the, I guess, assumptions that I feel like this maybe calls into question is in the non-coding genome. I think people have generally held a belief that it's going to be smaller effect size change, and we know that from GWAS studies. That's obviously largely true. But in the monogenic world, it'll be interesting to see if there are more cases like this where they explain 0.5% or 0.25% of all cases, because I don't think anyone would have predicted that you could find a single non-coding, not even just a non-coding element, but a single non-coding base change that would explain almost 0.5% of these cases. And I'm interested to see whether this is one of those exceptions that proves the rule, where maybe we don't really ever find anything that's quite this exceptional. But I think it would be more interesting if it was the other case, where there are other hot spots in the non-coding genome where maybe the mutation rate is very high, like it seems like it is here, but it's very high in a really critical part of the non-coding genome. And you get that perfect storm where you have both a very high mutation rate or a selection effect, like you alluded to. Maybe it's makes sperm more motile or more survivable or something like that, like we've seen in some coding regions that confer an evolutionary advantage in some contexts, but then result in a disorder. So I think it'll be interesting to see whether this becomes something that we see more often in the non-coding genome,
Yeah.
or whether this is just a really interesting edge case that's gonna help a lot of families, but won't necessarily be a new kind of class of things that we look for going forward.
Yeah, a similar estimate from the coding is like the maximum was 0.2%, I think, for the neurodevelopmental disorder caused by some gene called PAX1 or something. So it's, like almost more than double the estimate of that. And it's a really exceptional case.
Yeah, and probably worth saying that I did my PhD work in looking at a different set of non-coding regions. So I didn't look in this region specifically in the main paper that I published, but we looked at ultra-conserved non-coding elements. So we didn't find any smoking guns like this in that set of about a few hundred highly conserved regions that we looked at. But like you said at the beginning, the non-coding genome is a really big place, and we don't know. We certainly haven't explored all of it. So I think it'll be interesting to see how many more of these we come across. We've spoken about Parkinson's disease in previous podcast episodes, and people may be familiar with LRRK2, which is a specific genetic subtype of Parkinson's disease. But Veera put out a really great Substack post that we'll link to that talked about this interesting new paper that discovered an additional target or an additional piece of the puzzle in the LRRK2 pathway that I think is interesting for a variety of reasons. But I'll let you tee the story up here, and then we can talk a little bit about why this is interesting and eventful for the field.
Yeah, so this paper came out like maybe a couple of months ago. And so, I'm interested in neurodegenerative conditions. So when I saw this exome-wide association study of familial Parkinson's disease, I just started reading the paper. It was a new gene. It was interesting. And that kind of, took me through the rabbit hole of LRRK2 literature. I knew about LRRK2 before. I mean, like there is a drug being developed for Parkinson's by Tenali Therapeutics, but also a lot of other companies, I think, focusing on LRRK2. And but I never realized there is so much of fascinating literature in this. So I was like digging into that, and it was then I realized that like it's not just any other new gene being discovered. It's like it is very important gene, probably part of very important piece of the puzzle, the big block to puzzle that people, like scientists have been like putting together for a long time. So basically, the paper, there's two papers actually. So one of the first paper that actually brought my attention to it was published in Nature Genetics, and it was a exome association study of familial Parkinson's disease. And it seems that we did not, like most of the genetic studies of Parkinson's disease, the rare variant studies focused on sporadic or maybe sporadic and familial combined. And there was no, like proper at scale investigation of the genetics, of familial Parkinson's disease. All we know is based on isolated family studies, like a family based association analysis. And when they did this, I think they had like 2,200 cases and 70,000 controls. They found two known genes, GBA and then, LOG2. When at the same level of significance, they also find another gene, RAB32. It's a present in point, I think like 7% of the familial Parkinson's disease. That's really a big number, and we didn't know about that until now. And what is this gene? When I looked into it, that's when I realized that it's very much related to the log2. So a little bit on the, the log2 literature, right? So it's one of the most common, genetic cause for Parkinson's disease. It causes, it's seen both for familial Parkinson's disease and sporadic Parkinson's disease. And there are like many recurrent mutations in this gene. The gene itself was discovered in the early 2000s. So based on like we have this whole genetic locus that's mapped from the family based studies linkage analysis, and they identified the log2 gene in 2004, two parallel papers. And then so one first time when they identified the gene, so they didn't know anything about the gene. When they studying the structure of the gene, so they found there are like specific domains, and one of the domains is a kinase domain. So that led to the realization that probably the kinase function of this gene is what, like contributing to the pathology, the neurodegeneration and Parkinson's disease. And I think this is the moment when the whole cell signaling field turned their heads towards Parkinson's disease, because it's a kinase protein. So and it's like they found it's a kind of a like gain of function mutation. So it doesn't matter. You can like you can develop drugs to target the kinase protein. That's the whole thing in the cancer field, right? So I think everyone, a lot of people jumped into this and started working on it. But it surprisingly, it turned out to be very complicated than everyone expected. And so the lot of mutations are clustered in the kinase domain. And so initially they realize it's a kinase protein. It also has a GTPase domain. There are also other domains. And so after they found out it's a kinase, protein kinase, meaning it can add a phosphate group to itself or to another protein. And so the one of the main question that turned out natural question was like, what is the natural substrate of log2? It looked like a very simple question, right? But I think like it took 10 years for the field to actually identify the target through like meticulous in vitro, in vivo animal model experiments, trying to knock out or activate log2 or knock in specific log2 mutation, and study the phosphorylation profile of hundreds of proteins in the brain tissues or in the cell lines. And finally, they found there is like a family of proteins called RAB proteins. RAB stands for ROS-like proteins in the brain, and they are highly expressed in the brain, all these proteins. So they are the natural substrates. So that's one main puzzle they solved, and then they tried to understand how the log protein actually activates or phosphorylates RAB proteins, and what happens after that. there is like dozens of RAB proteins it is phosphorylating. Which one is contributing exactly, causally to the neurodegeneration? I don't think we still know, but people are trying to understand the consequence of each of the RAB proteins that the LOCK2 phosphorylates. But the other most interesting question that came up is because LOCK2 is mainly present in the cytoplasm, and these RAB proteins, right? They're all actually present near the membranes. They're present in the plasma membrane, also in the membranes of the cell organelles, endoplasmic reticulum, the lysosomes. So something has to, pull this LOCK2 from the cytoplasm to the membrane. So at any time point, less than 10% of the LOCK2 is seen in the membrane. When it is seen in the membrane, it is activated. You have a lot of, you can measure the activation in terms of phosphorylation. Rest of the LOCK2 is present in cytosol. So the question, so they wanted to solve is like how LOCK2 is like brought to the membrane or the mechanism of activation. So that is very important because you need to know how the LOCK2 activation is happening. And if you want to develop drugs to reduce the activation or inhibit the activation. And there's like a couple of proteins they identified, one through functional studies like RAB29, but there is also some link with the genetics. It's sitting in one of the Parkinson's locus identified through genome-wide association study. And there's another gene called VPS35. It was identified through genetic studies, like mutations in this VPS35 actually causes highly penetrant autosomal dominant Parkinson's disease. And they realized that it is actually in patients with these mutations in the blood cells, LOCK2 actually is like activated, overly activated. And they can also show that in the cell line. So this is all the literature happening, right? So I'm talking about studies that's happening for like probably 20 years since the discovery of LOCK2. And none of the studies actually got anywhere close to RAB32, right? So, and then now looking at the genetics, and then it says, hey, here is a gene. Probably, so they've found evidence that it is actually interacting. Already there is a evidence that it's directly interacting with LOCK32 and it is activated. It is humbling, right? I mean, this is what is like another example of no level of, like all kind of this functional experiments or anything can equate with what we can learn from natural experiments. And so this is like really an example of that. And this is really exciting because now nature has threw a clue to the people who've been working on this for 20 years on solving the LOCK2 puzzle. Now already people have started probably working on how the mechanism works, right? So here it seems this is also a gain of function mutation. This is a missense mutation. There's they identified just one mutation is contributing to this association, and it is located in the only phosphorylation site in the RAB32. And so the lot of questions right now is like, okay, so if it is related to LOCK32, like there's already LOCK2 inhibitors are currently in development. So will LOCK2 inhibition work in patients with Parkinson's disease caused by RAB32 mutation? Or, if RAB32 might be a better target than LOCK2, right? So if RAB32 is in the causal pathway to the neurodegeneration, right? So a lot of interesting questions to answer. But the overall, thing for me is that like how the human genetics is like highlighting this one finding that people did not catch it using all these kind of experiments for a long time. And that's what like really makes the story super interesting. And also reassures us, this is the way to go. You cannot replace human genetics with anything else. So the only way to go is to collect the samples, do the studies, do the association studies. We are nowhere near saturation, particularly for neurodegeneration disorders. We are just beginning to understand all these risk factors. So I think it's one of the incredible papers of this year.
Do you think, because when I looked at the paper, the thing that stands out is so the Manhattan plot has 3 hits, GBA and LRRK2, which were known, and then RAB32, which is the new one. The reason why RAB32 is new and GBA and LRRK2 have been known for a really long time, it's probably not a power issue, or is it? Because the reason I asked the question is I'm wondering how many more of these genes we should expect to find, and what's the limiting factor? Because in some of these stories that we have, it's a new population that we found this because we had sequenced only mainly Europeans before, but then we sequenced a new population and boom, a brand new gene arrives. So what's the story in this case? Why do you think it's taken so long to discover RAB32 when it's not, like you said before, it's close to 1%. It's about 0.7% of the familial cases. So it's not, it's not an insignificant number. What do you think is driving that?
Yeah, so I think power issues are one of the things. And like I said in the starting, that familial Parkinson's disease in isolation have been not studied for, genetic studies. So there is huge heterogeneity in the genetic risk factors between familial cases and also sporadic cases. So probably when you combine them, you dilute it, but also, so you lose sight of some of the important genes. So it's 7% among the familial PD cases, but it is probably only half of that, or even lesser than that when you focus on sporadic cases, and that is why people probably have missed it. And I'm sure now everyone who have their own cohort of familiar Parkinson's disease probably start looking into this particular gene. And so I forgot to mention the parallel paper. So the parallel paper was published in Lancet Neurology, and in that paper, also they identified RAB32 in a substantially lower sample size. Like in this year, 2,000 cases. I think they have probably 160 cases or so. But they used a hypothesis-based approach, and I think this is also important because all this years of functional studies have helped those scientists to actually find the RAB32 at the same time, right? So they hypothesized that, okay, we know RAB proteins are the natural substrates of LOG2. So let's look into only the RAB proteins. So there's 70 RAB proteins. So they looked into the RAB variants and across the RAB proteins in 160 familial Parkinson's cases, and probably some 200, 300 controls. And they found that the strongest variant was this particular serine 71 or G9 mutation in RAB32. And it was not in terms of significance were comparable to what was found in the Nature Genetics paper, but they found it. So that's another way to identify risk factors. I think the combination of these two is really important because our next discussion will be focusing on hypothesis-based, finding of genetic risk factors. And so I think if we want, this is really important if we want to accelerate the discovery. So there is one group who constantly criticize candidate gene studies, because it's understandably abused in the GWAS literature, right? So, and then so they're extremely strict in terms of what they consider as significant, and they are extreme frequentists, and they focus on p-values, and they will believe only when you have large scale study. But for conditions that are rare, with huge heterogeneity, that is shouldn't be the only focus. So I think it's really, really important that we also on the side try to accumulate whatever the functional knowledge, the biological knowledge that we learn from initial discoveries to apply to accelerate the discovery. And we have an example on our next paper on that.
Yeah, that's a great segue. Let's go right there. Maybe you can talk about this pediatric lupus story, because and it's a good continuation from one of the ones we discussed a while back.
Yeah. So here, so this is a paper was published in Nature Immunology a few weeks ago, and I was preparing for was looking into this paper again for our podcast, and I realized that there's two other papers that also reported the same discovery. So here the discovery is that they found that there is few mutations in a protein, a gene called UNC93B1. That is as a rare monogenic kind of risk factor for SLE. I mean, this goes into the TLR, toll-like receptor literature. So I think I started, I looked at I came across a paper 2 years ago that reported a gain of function mutation in TLR7 that as one of the rare monogenic cause of systemic lupus erythematosus, early onset SLE. I think that is my entry point into really digging into the literature on the TLR biology and its relationship to, autoimmune diseases. it's really interesting. I mean, like, when you look into the how this all came up, like SLE is an autoimmune condition. And one of the characteristic feature of SLE is like, the patients develop antibody against their own nucleic DNAs. Anti-nuclear antibodies is the characteristic of SLE. So they develop, they are antibodies against the cells of all different parts of the body. So the patient's skin is affected, kidney is affected, blood is affected, like sometimes also like brain tissues. And so we know about SLE for a long time, but the TLR7 biology came up, I think, in the early 2000s, when the breakthrough discovery happened with in terms of identifying the pathways that actually sense the like viral and bacteria derived RNA and DNA in our body, right? So that happened in early 2000. They identified the toll-like receptors, intracellular toll-like receptors. So like there are TLR, 3, then 7 and 8 and 9. So they all sit in the inside the cell in the endosomes, and they sense different types of nucleic acids. Like they sense single-stranded RNA, double-stranded RNA, also like like unmethylated DNA. A lot of this come from pathogens. And so when those breakthrough discovery happened, that's when the realization happened. Okay, then this has to do something with the SLE. Like we always know, the autoimmune disease and the infection lie in a spectrum on the opposite end of the spectrum. On one side, like extreme immune response causes autoimmunity, and poor immune response causes, like immunodeficiency.
Yeah.
So that's when they discovered this, like they started the intuition happened like, okay, TLR7, TLR8, these should be related to SLE. The very interesting part of this is that, we know SLE is like extremely common in females, like 9:1 ratio, I think. So what is the reason for extremely high common? So the natural thinking would be to X chromosome, right? And it turned out to be true, because these genes, the TLR7 and 8, are actually located in the X chromosome. That was their initial clue. So then there's like in very interesting mice. There's like many autoimmune animal models, but one spontaneous model came up when they actually tried to cross between two breeds, and then that developed, like spontaneously developed SLE. And when they actually investigated what is actually causing this the SLA, they found that there is like, they are looking at the like the chromosome, and they found there's like a piece of the X chromosome is actually sitting inside the Y chromosome in this animal, and that actually was carrying the TLR7 TLR7 gene. It's known for this nucleic acid sensing. So that is one of the very first evidence to show that TLR7 overactivation, right? So now there's like two copies of TLR7 in this mice, or it's causing like over functioning. The gain of function causes autoimmunity. And so then there's like so much work went into that, but there was never a human genetic finding, a naturally occurring gain of function mutation in TLR7 causing, SLE. And that happened 2 years ago, 2022. It was just one child, like they found in one child with the early onset SLE. And it is just knowledge of all this actually led them to go and look for this mutation in TLR7. It kind of, for me, reminded me of how obesity mutations were discovered. The first leptin gene mutations were discovered from mice, from naturally occurring OB mice in the Jackson laboratory. And when people with extreme obesity was like coming to clinic, and patients, like scientists like Stephen O'Reilly or other people, like Gail CEO, were like looking specifically on this gene that actually causes this mutation. So this knowledge actually helped to discover these genes. It's a very similar story here. They found this TLR7, right? So now they started, the starting point is toll-like receptor. Now, over the years, people are starting to paint the picture of the pathways, what is happening with TLR7, how it is causing the SLE, what other genes are there. And one such gene is this E1C93B1. It also they found through my study, where they did use this mutagen to create random mutations. So one of the mutations was a mutation in an unknown gene. They didn't know what is the function, but that kind of caused all toll-like receptor related signaling defects and causing immunodeficiency. And then that led to the discovery of this protein. It is a chaperone protein. It traffics all this TLR proteins, after they are produced from the endoplasmic reticulum to the endosomes where they actually are. So when you have some defects in this chaperone protein, it also results in TLR signaling mediated diseases. One of this is SLE. And then that is how led to the, the understanding of UNC93B1. There's crystallographic studies, there are a lot of mice studies, there's like few mutations. But until this year, 2024, I don't think there was no examples of genetic risk factors, naturally occurring mutations in this causing, SLE. And now, like not one, but three teams of scientists, one from the China, and the other from the INSERM Institute in Paris, and also there is another research group from University of Dresden, Germany. All of them actually, I have encountered, these patients with early onset SLE where they found mutations, different kind of mutations in this gene causing, early onset SLE. So they did some functional experiments. They found that actually the TLR7 signaling is specifically, like enhanced. There is like a hyperactivation of TLR7 when you actually put an agonist for the TLR7 in the cells. And so this kind of, proves, like kind of agrees with what has been researched so far. And the very important aspect of this is this has a very clear therapeutic implications, because TLR7, we have antagonists already in the market, is be used for different indications. So probably TLR7 antagonists can be used for the treatment of this specific risk factor for SLE. And one of this paper, the Nature Immunology paper from the researchers in China, so they found there's one particular mutation that is actually seen only in the Chinese population. There seemed to be a variation in the allele frequency across the latitude, and with the highest concentration seen in the southeastern part of the China. And this is also really important because now you have a therapeutic that is probably more important in a particular population, and so in kind of a personalized medicine, right? So I think that is like really interesting because now we have at least, like already existing drug that can be repurposed for this indication. And probably it will be more curative than the standard, treatment that is existing, currently used in the treatment for SLE. And yeah, so for a lot of these reasons, it's really interesting. And I think it's impressive that, like there's a big gap, no one find anything. And then suddenly, like you said, like 2 papers, 3 papers, everyone
Yes.
starts finding. It's just like force down all these discoveries. That always impresses me. Yeah, everything that I see in the literature is always like that. There's like parallel publications, people arriving at the same finding at the same time.
Yeah, it is. It's interesting. I mean, I think it speaks to what a competitive and like kind of liquid market it is. And the first paper we talked about, actually, there was a very similar finding published just a couple of months afterwards. That presumably they were looking at the same thing at the same time, and one of them went to preprint first, and the other one went to a peer-reviewed publication first. So it is very competitive, and also there's a lot of dynamics that happen where as soon as someone catches wind that the story is being put together at a conference, then often there's a little bit of a rush behind the scenes. Who knows what happened in this lupus case? But often there's groups there's the same technology and same data sets, and they're kind of moving towards the same ideas at the same time.
In that case, it's the same data set. Like the both papers looked into the genomics England data set.
Yeah.
But here it's 3 independent data sets from independent populations.
Interesting.
It's like really super interesting. And none of them are like big samples, they're all like 5 patients, 6 patients, and they wouldn't have found it if they didn't know about this gene before. So that prior knowledge, biological knowledge, is really fueled these discoveries. And I think that's like really important, and that people need to appreciate it. I mean, there is, like I mentioned before, there is a current trend. The data sets are getting bigger. So, like you can do analysis in millions of individuals, or, like hundreds of thousands of individuals. That shouldn't make you, like see the other approaches, like it's not useful. I think these kind of papers really reminds us about how we should still anchor to the biology if you want to really make meaningful discoveries.
Yeah, definitely. On both the Parkinson story and this one, I don't have any papers to point to on either of these, but one of the things that I'm interested in seeing how it might develop over the next couple of years is where we see these monogenic familial studies or pediatric kind of early onset studies colliding with the polygenic risk score body of work. Because I've got to imagine, if we take LRRK2, for example, there's going to be people approaching this question from the polygenic risk angle to say, are there, how does the kind of GWAS literature and all the work that's being done there that points to smaller perturbations along the LRRK2 pathway that eventually kind of cause a much higher risk of Parkinson's disease, but via several much smaller changes.
Yeah,
and start to look at these monogenic changes. I think there's going to be something interesting when both these data sets grow in size, that you're going to start to see, either a convergence on the same pathways and same genes, or in some cases, maybe there's a divergence where you've got really truly familial or monogenic cases that are somehow fundamentally different from the more polygenic driven ones. And I just think it'll be interesting to see how those play out and whether these converge or diverge depending on the disease.
Yeah, I think that's very, very important because, like when the LRRK2 thing, they found that there is also, GWAS risk locus. And, initially, probably they thought this kind of a monogenic conditions, right? So probably any therapeutics that you target LRRK2 probably will work in individuals who have LRRK2 mutation and that causing the Parkinson's disease, but not in other, Parkinson's disease patients. But the finding that LRRK2 comes up as genome-wide significant locus, and there's like mild perturbations in the LRRK2 actually increases the risk for Parkinson's, kind of puts LRRK2 as a important protein in the biology of Parkinson's, neurodegeneration biology. So that kind of makes your target population bigger, right? So probably any therapeutics again to LRRK2 is not just indicated for LRRK2, but also probably for other Parkinson's disease because of the importance of this gene. And yeah, I think that probably that is also a same line that in our next paper, maybe we have like few minutes. I guess like we should try to
we should
quickly summarize the next paper.
Yeah, this was the one that we wanted to make sure we got to, but realized that we probably wouldn't have as much time as some of the others. So we did plan for this to be a quick one. But yeah, it'd be great to hear your thoughts on this really interesting paper.
So just to keep the continuation of our Alzheimer's, we've been discussing almost at every podcast now. So we discussed about ApoE targeting, how naturally, occurring genetic mutations are reassuring, okay, targeting ApoE probably like a good therapeutic approach for Alzheimer's treatment. So we saw this paper for ApoE truncating mutations, like removing the effect of ApoE4, like Alzheimer's causing mutation. So validating that, like inhibiting the ApoE as a therapeutic approach. And so like similarly, we also discussed about this one Christchurch mutation that actually protected an early onset genetic risk factor SE, and one mutation that is seen incredibly, like many patients in the Columbia, Antioquia place, right? So that was one of the biggest finding. Was like, I think 2 years ago or 3 years ago, was published in Nature Medicine. They found this one woman who did not develop cognitive impairment until like her 70s. And so even after, I think she died, they did postmortem examination and they did not find any pathology that are like specific for Alzheimer's, except for a high amyloid beta burden. They found very low tau burden biomarker for neurodegeneration, and also they did not find this amyloid angiopathy, and so that was like really interesting, but it was just n equal to 1 case report. But people have been like, now slowly looking into that's an homozygous Christchurch mutation. So the question was like, what happens in people with heterozygous Christchurch mutation, particularly people with PSEN mutation who also carry one copy of the Christchurch mutation? What happens? They did not have, sample size at that point. Now they have collected enough sample size. I have like 27 patients. They followed up and they found the clinical profile. And it turns out there is like a moderately, like a moderate effect size protective effect is seen. The age of onset of cognitive impairment. So the people with PSA1, PSEN mutation develop cognitive impairment, mild cognitive impairment in the early 40s and dementia in the late 40s around, And so here, people who carry this protective, mutation, one copy of Christchurch mutation, their cognitive impairment is delayed until post 50, age 50, like 5 years on average. And so that's kind of, like sits in with the extreme protection seen with the homozygous mutation. And also they did this also imaging studies, which showed actually very low tau burden and also healthy, like metabolic profile, preserved metabolic profile. And interestingly, like how they found the high amyloid beta burden in the patient, in that one patient before, right, in the early report. Here also they find there is like relatively high amyloid beta burden. And despite this high amyloid beta burden, these people are having, like a delayed onset of cognitive impairment. And this is like really interesting because now it says one thing that is targeting ApoE is like we're getting more and more evidence that ApoE targeting probably is a good approach to treat Alzheimer's disease. And the other thing is like it kind of decouples the amyloid beta thing from, like the current understanding or the, like the angle towards we go is like kind of reduce the amyloid beta burden to treat the Alzheimer's disease. But it's kind of show that, even if you don't reduce the amyloid beta burden, probably there is some other part of the biology that you can target probably by, inhibiting ApoE or something. And that could, still can have like a therapeutic effect, even if it doesn't reduce the amyloid beta burden. But it's just speculation. But it's like really interesting that they find this, heterozygotes, 27 carriers, and this really adds to the homozygote function. I think this is like very important advancement in the our understanding of Alzheimer's, particularly in terms of ApoE therapeutics. And so I just wanted to put that in our list today.
Yeah, it's very interesting. And there's a related but kind of totally different thread that I think is worth people following on this, which is there's a phase 3 clinical trial that I think is going to read out any week or month now. So there's a biotech company called Alzion that I've mentioned a few times before, but they have had a phase 3 trial called Apollo E4 that's for E4, so homozygotes for ApoE4. And they fully enrolled. I think, based on all the public reports I've seen, they're analyzing the data as we speak. And I think they had good data that came out of the phase 2. And so if the phase 3 data looks good, we could see potentially the first treatment for ApoE4 for homozygotes. And I think it will provide some really interesting clinical data around. They have a small molecule that's really kind of targeted around this mechanism. And I think the results, positive or negative from that, will tell us something really interesting about ApoE4 as a target.
Yeah, what happens?
Yeah. So I don't know. I'll definitely try to get somebody from that team on either way once we see what the result of that trial is. But I think it's particularly interesting as well because we know that now there are multiple approved Alzheimer's therapies with Lilly's donanemab getting the nod just today, I think. But we also know that in most of these cases, there's significantly higher risks of ARIA, brain bleeding, in people who are 4/4 homozygotes. So there's clearly something going on here. And there's a paper recently that I think suggested that the 4/4 carriers really kind of, there's strong evidence to suggest that they should be considered almost a different molecular pathology and if we go so far as to say a different disease or subtype of disease. So it feels like all these things are coming together, and over the next couple of years we should get a clearer picture of whether this is one disease with multiple different genetic perturbations that lead to the same thing, or whether we really need to think of them as different diseases or different, at least, molecular pathologies that are, creating similar symptoms, but really shouldn't be treated or thought of the same way. So I think I agree with you. I think it's a super interesting story, and one that I'm probably going to read the paper again now that I've heard you talk through it, because I think I've learned a lot and have just thought about it a little bit of a different way, hearing your perspective on it, as I always do.
One aspect of this that I really, like kind of learning point here, for me at least, is like, so I title this as like new insights on old genes. We do genetic studies, we discover some genes, and then when we do the next phase of the study, bigger sample size, like I always get frustrated when reading GWAS papers when people say like, okay, 3 are known genes, okay, and then that's it. Just they focus on all the novel genes, but it doesn't matter if it's a known gene. We didn't know anything about it. What new finding we know about it? Like it's kind of there is a tendency to ignore known genes. Like it's really important that one of the important thing as we increase the sample size and, do studies is to find new insights on, like how we discover genes. And so these are the studies to, showcase that example of there is still so much we can learn from all these known genes by increasing the sample size, identifying this, rare set of carriers. I think this kind of approach is really important. And
couldn't agree more. So that wraps our 4 stories. I just want to say thank you again for doing this. I always enjoy these. I always learn a lot reading the papers beforehand and also talking through them with you. Ahead of the next one, if any, we do these every 3 months. So it's aligned with I didn't even know what a quarter was when I was a scientist, but you probably didn't either. But now that we work in industry, we know what quarters are. So we do it aligned.
Q1, Q2.
Quarters. Yeah. So this will be our Q2 roundup. So around September time, if there are papers or findings that you think are really interesting that you want to send our way and help put it on the list, then we'd love to hear.
Yeah, already there's a few exciting papers in the list.
Yeah. And we're only 3 days in. We're recording this on July 3rd. So I hope if you're going to take some time off for July 4th, that you go to barbecue, enjoy yourself, take some time off. And I'll definitely see you 3 months from now, but I'm sure we'll see each other sooner.
Thank you very much for the time and invitation.
Yeah, pleasure is mine as always. We'll see you all soon. Thanks everybody for listening. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.